All the other material has been cleared, tumors are lighting up. And these are systemic tail vein injection. We're not injecting into the tumor. This is one injection into the tail vein, and over time it accumulates. Because you can see as you go back, you can see the tumors as early as 24 hours. The other thing you see is, look very quickly, you start seeing the material, that's the liver. The material goes to the liver, but doesn't accumulate in the liver. And because of size, it actually ends up going through the hepatic biliary recirculatory tree, and you can see over time we've actually pulled out the intestine, and we've actually published this, but the material ends up in the poop, in the feces. So this is a nice way to get rid of the material. Nice clearance mechanism, and it does lead to minimal liver toxicology, and only 5% of the material goes to the renal system anyway. Okay, so now we can actually see the tumor by enhanced permeability retention. Here's one more example, another animal, again a nice example of the only thing lighting up is the tumor. We've then taken the calcium phosphate nanoparticles and we've put in gold. And now we use EM to look at the gold calcium phosphate nanoparticles. And you can see, you might have turned down the lights, but you can see the gold encapsulated calcium phosphate nanoparticles can be picked up. They're not agglomerating, they're in the endosome, and that's where they're going to dissolve. And that's an example, one of the proofs we have that these materials can deliver cargo by this pH reduction. This is what we call the Costco experiment. We actually went to Costco, one of the large purveyors in the United States of food, and we bought a pork butt. And basically, we actually put in the indocyanin green particles, either free indocyanin green and or indocyanin green within the calcium phosphate nanomatrix. And we can see in this example, you can see up to 3 centimeters through biological tissue. In the experiments we're now publishing right now, we're going 6 centimeters. So that's talking mammography, we're talking the ability to image through biological tissues. And the reason we can do it is because we get this quantum efficiency, this increased bang for the buck, because we've encapsulated within the calcium phosphate matrix. That's all passive targeting. This is actually going to be now published in a week and a half in ACS Nano, again with an editorial. This is now, can we actively target the calcium phosphate nanoparticles to the tumor? So what we've done here is we've done it 2 ways. The left side of panel B is a strep avid and biotin mechanism to put an active agent on the outside. The right animal here is we're using a maleimide chemistry. And what we're doing here is we're either putting pentagastrin or decagastrin and coating the outside with the 5 or 10 amino acids of the gastrin molecule. Why? Because some of our collaborators, such as Jill Smith, who's an oncologist, has shown that pancreatic cancer, but not normal pancreas tissue, expresses and synthesizes and releases gastrin and has upregulation of gastrin receptors, particular to a specific small part of the gastrin molecule. So what we're seeing right here, and you can see really here at 24 hours, that spot, that's the pancreas. When the material was not actively coated with the active targeting moiety, the pancreas doesn't light up. When you put the gastrin on the outside through bioconjugation, you now light up the pancreas. There's the pancreas, that's the pancreas. These are the stomach too. That also expresses some of the gastrin receptors. So we can actually now move the nanoparticle to the tissue we want to go to. So let me spend the last 5-6 minutes on what we can do with this. This is all about imaging. But I said we want to be a theranostic. We want to do therapy and we want to do imaging at the same time. So let's go into what's called photodynamic therapy. And this is one of those serendipity type of experiments that basically go, I'm glad sometimes people make mistakes. So Brian Barth, who's one of my senior postdocs, basically came to me one day and said, Dr. Kester, Mark, you've got good news and bad news. I said, well, okay, what's the bad news? Well, we usually do experiments, as you saw, for 96 hours. And then we just want to do the imaging, and then we sacrifice the animals. And we have multiple animals experiments going on in real time. He said, well, we forgot one series of animals, and basically they went on for a month and a half. And you got to pay the bill. I said, what's the good news? They don't have tumors. So that we started thinking, can we use this enhanced quantum efficiency of the light to actually kill tumors? The technology is known as photodynamic therapy. It's used for esophageal cancer, bladder cancer. It uses a photosensitizer, light, and oxygen. The photosensitizer is excited, and basically you form a single molecular oxygen species, which then is activated in the single oxygen state and makes reactive oxygen species, and you kill the tumor from the inside. The problem with this therapy is basically the photosensitizers don't work. They have very low quantum efficiencies or quantum yields. The short half-lives are in the order of minutes. You have lack of specific targeting, inability to penetrate the targets, and basically the mechanisms are not well worked out. And what's not here, these tumors are hypoxic for the most part anyway, so you're not going to have much oxygen to make reactive oxygen species. What we're banking on is the fact that because we got the quantum efficiencies, because we got the protection, because we got the increased half-life, because we can target, maybe we can resurrect photodynamic therapy. And in that process, we came up with photoimmunonanotherapy because we see we're going to be working through a whole new mechanism of how these photosynthesizers work when they're encapsulated. So let me just go right to the chase. Does it work? So here we have two different mouse models. This is a nude mouse model. It's a breast cancer tumor. It's an MDA, highly aggressive estrogen-responsive tumor put into the hind flank of the animal. Here we have a syngeneic model. We're putting a mouse tumor inside a mouse with a complete immune system. We'll come back to that in important. I just want to go down to this green line and this green line over here. What we're doing is we're taking the calcium phosphate nanoparticle with the indocyanin green encapsulated. We stick a bolus into the tail vein for 24 hours. We see within 24 hours that the material ends up in the tumor. At that point, we take a very good photo laser and we actually shine it at the tumor. And we can actually focus the laser. If it's an orthotopic model, we can focus it inside the animal. If it's on a nude mouse back, we can focus it on the tumor. So it's one injection, one shot of the light. And then we follow the tumor over time. Here are all the controls: PBS, ghost particles, free indocyanin grain, calcium phosphate nanoparticles with indocyanin green but not have the high pegylation. All the tumor is growing. This is the indocyanin green calcium phosphate nanoparticle, highly pegylated. Tumor doesn't grow. All in the nude mouse model, but also in the syngeneic model. Bottom line, we made a theranostic: image and treat. We actually went, that's a solid tumor. We even made it harder. We said, well, can we do it in a non-solid tumor model? This is a model of leukemia. It's a CML model. It's basically, we've extended the lifespan of this animal about 30%. And here we're actually focusing the laser on the spleen, where we're actually getting a lot of the leukemic cells being formed. Again, here we're giving 3 IV injections, and then we're giving 1 treatment with light, and we can see we extend that lifespan about 30%. How does it work? So this is, again, that in vitro proof after the in vivo. So actually we saw that the indocyanin green calcium phosphate nanoparticles can reduce in a cell viability assay about 30%, 20-25% of the growth of these breast cancer cells in culture. And we add in an inhibitor of reactive oxygen species, basically tumors grow very nicely. So oxygen is important, but it's not the only mechanism. And here we're actually looking at energy density, energy yields as a function of the nanoparticle. Here's the encapsulated material. Here's the free indocyanin green. And again, as a function of concentration, we see increased energy density, energy flux as a function of encapsulation. So how is it really working? And this is where we get into the photoimmunonanotherapy. So there are myeloid depressor cells which basically reduce the activity of the immune system and are downregulated in, are upregulated in certain cancers. So you don't have the immune system attacking your cancer. So here we've used 7 color flows cytometry, and basically we've come up with 2 different subpopulations of myeloid-derived suppressor cells and kind of look here versus here or here versus here. There's less orange here than these upper 2 panels. This is the indocyanin green calcium phosphate nanoparticle. We have less myeloid-derived suppressor cells. At the same time, this is in the nude mouse model where there's still an active T cell component. We also see the same phenomenon in the BALB/C mice, decreased myeloid-derived suppressor cells. And at the same time, we're seeing an upregulation of natural killer cells, and you'll see the next slide, dendritic cells.